Today, we're going to learn about the DNA in the cell cycle. It's Dr. Snuggie here, and we're going to start off by learning about the discovery of DNA. So first, Frederick Griffith, in his experiment, he killed virulent bacteria into a mouse, and then he found out that this transformed avirulent bacteria into virulent ones. And then Hershey and Chase found out that DNA was in fact the infecting agent that carried the bacteriophage's infection from the virus in its DNA. And then later on, Crick and Watson discovered that using Franklin's X-ray crystallography, that DNA did have a double helical structure, and that the purines were paired up with the pyrimidines, A to T and C to G.
Now, moving on to the replication of DNA. DNA has a double helix structure. Nucleotides bond from
Phosphate
and sugar.
hydrogen bonds, which are weaker,
Hey!
between the nucleotides and covalent phosphodiester bonds, which are stronger, between the sugars and phosphates. Helicase unwinds the DNA into two single strands and uses the single-stranded binding proteins to stabilize the DNA strands. Topoisomerase keeps the DNA strands from rewinding back together.
No, stay apart.
DNA polymerase III is a main enzyme of replication, proofreading, and editing, as it is involved in the prokaryotic DNA replication. It builds the new daughter strand and adds new complementary bases.
Go away, I'm doing stuff, I'm building right now.
Okazaki fragments become the lagging strand, and it is created from 5' to 3'.
Okazaki is always lagging. And why is it called 5' to 3'? I mean, I'm building upon the 3. That's where it should start, right? I don't get why the bigger guy always wins. Oh, I'm DNA polymerase. Nice to meet you.
Rashu, what's wrong?
It's been a rough day in the mitosis factory.
I'm so sorry, man.
Well, in the mitosis factory today, Dan screwed up. So now instead of 2 sister chromatids, we have 3.
Kevin, I told you it's like this.
Dang it.
Kevin is so exhausting. Even the little girls know how to do it. Come, I'll show you.
Interphase, prophase, metaphase, anaphase, telophase, /cytokinesis. Cell division needs to be regulated across tissues because it's crucial for normal growth. However, at this factory, no one knows how to do anything. The 3 major checkpoints, G1, G2, and the M phase, none of them know how to do their job.
I'm dividing.
G2, nice to meet you.
Nice
How was
to
your
meet
day?
you. Great.
You're doing great. Go ahead. Okay.
Thanks.
Actually, come here. Look what I mean. Look at, she has a pencil sticking in her hair. Really, G2? Really?
You're here too? You camera people are always following us around. Oh, well, she always complains about me doing my job, but she doesn't understand that I'm the one who gives the signals the green lights to go because I do the external and internal processing.
Meiosis is just like mitosis, but it's sex.
Go away, Dan. You're so inappropriate.
Meiosis comes in 2 parts, meiosis 1 and meiosis 2. Pretend like this is a cell, except it's not. It's actually a carrot because we're vegan. Anyway, so in the first division, there's 2 cells in meiosis 1. In meiosis 2, there's no interphase, but then each cell divides again into 4 cells, each genetically unique.
Welcome back to the factory. So now let's talk about cancer. Cancer is basically your body failing to control cell division. You know how earlier I was telling you about how Dan can't control the checkpoints? Well, in cancer, the checkpoints stop working. So basically, Dan's causing cancer. Growth factors create cancer. One of them is proto-oncogenes. They activate cell division and become oncogenes, which means cancer-causing when mutated. When it's switched on, it causes cancer. Tumor suppressor genes inhibit cell division, and if they're switched off, they can cause cancer.
Tumors are massive abnormal cells. Benign tumors start at the original site, and they just stay there. And then malignant tumors start from the original site and travel to everywhere across the body. Hey, Dr. Snuggie back here to talk to you guys about the best way to help fight cancer. Chemotherapy. Not only does it stop blood vessel growth, it also stops mitosis, cytokinesis, and cell replication.